All right, so we know about DNA's two roles. It's got to be able to copy itself so that offspring can get copies of the DNA so they also know how to work. But DNA also has to be instructions for creating living things, this protein stuff that life is made of. DNA is the directions for making that. And we've talked about transcription, about making the RNA middleman that is going to kind of help us get that DNA message out to where it needs to go in the cytoplasm. But now we're going to talk about translation, the actual making of the protein. Okay, just because we've got RNA doesn't mean we got protein, but it's an important step to getting there. Okay, so that's where we're going in this podcast. Okay, so we're going to get from the nucleic acid language to the amino acid language, which is the language of proteins. Please don't forget the central dogma. This is really important. DNA, which we know can replicate itself, but that's not what we're talking about here, is transcribed into RNA, which is then translated into protein, which is what gives us all of our traits. Okay? We're going to focus on translation. The players we're going to be involved with are the ribosomes out in the cytoplasm that cling to the endoplasmic reticulum, and also some of them there's float free in the cytoplasm. RNA called mRNA or messenger RNA, and a type of RNA called tRNA or transfer RNA, and of course the amino acids. Amino acids, if you'll recall, are the monomer building blocks of proteins. You can't build a protein without the amino acids. That's what builds proteins. All right. Let's talk about ribosomes. This is the place where we're going to make the proteins. They're kind of the factory where all this is going to go down. Two things must be brought to the factory. The instructions from the DNA, which we are going to see take the form of RNA. So we're going to bring the RNA to the ribosome, and we're going to bring the amino acid, the raw materials, the building blocks needed to build the protein to the ribosome. So what we're going to get is something that looks kind of like this. The ribosome is in two pieces, a big subunit and a smaller subunit. And the mRNA molecule that carries the genetic message from the DNA is going to be kind of sandwiched in between them. This big unit is going to come down and slam on top of that small unit, and we're going to have an mRNA sandwich. We'll talk about what these things are later. Don't worry about those now. Now, please remember, the amino acids have to be brought to the ribosome. There are 20 different amino acids, and much like the letters of our alphabet, we can assemble these amino acids in different orders to make extremely varied, complex structures. Just like these Legos comes in many different shapes and sizes, and we can put them together in many different ways to make many different structures. Same goes for our amino acids. We've got all these different ones, glycine, alanine, valine, and we put them together, and depending on how we put them together, we get many, many different proteins that do many different things. But if I'm going to build a complex structure, if I'm going to build a castle with my Legos, if I'm going to build a complex enzyme with my amino acids, I have to know what amino acid goes where. How will I know that? The genetic code is how your cells will know that. The genetic code is what is being carried by the mRNA from that DNA, and it is based on the order of the nucleotides that was in that DNA and now is being preserved in that RNA molecule. Okay? The genetic code, we have a chart that shows us what the genetic code is. The same code is used for all living things, which is really strong evidence that all life has a common origin because all life is using the same code to code for proteins. So, mRNA is carrying this genetic message from the DNA in the form of codons. What is a codon? It is a little 3 nucleotide word, uracil, 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 or cytosine, uracil, uracil, or adenine, uracil, uracil, that is telling us what amino acid, isoleucine, leucine, phenylalanine, what amino acid is supposed to go at this point in the construction of the protein. Okay, so 3 nucleotides, UUU, UUC, whatever, are required to code for 1 amino acid. You got to have 3 to tell me what amino acid to pick. It's called a triplet code for that reason. There are 64 total code words, codons. They are called codons, UUA, UUG, CUU, all those are codons, and there's 64 of them. There are not 64 amino acids. There's only 20 amino acids. So that means that some amino acids have more than one codon that codes for them. Okay? So, as I said, each different codon codes for a different amino acid. UUU codes for phenylalanine. But if you'll notice, so does UUC. So we have some repetition here, but nonetheless, UUC will never code for anything but phenylalanine, although phenylalanine can be also coded for by UUU. Okay? There are some special codons. There is a start codon that tells us we are at the beginning of the genetic message, and there are stop codons that tell us when to stop reading that genetic message in the mRNA, and they don't code for any amino acid. The start codon always codes for an amino acid called methionine, or MET. So MET will always be the first amino acid in any protein. Now, there is another type of RNA that is going to play a role besides mRNA, and it's tRNA. And remember, the T stands for transfer. It could also stand for tow truck, because it is towing the little amino acids around, and it's going to bring them to the ribosome. Okay? It's going to bring each little Lego to the ribosome. There are many tRNAs floating about, and each tRNA molecule is carrying around 1 amino acid, one of the 20 different ones that exist. Okay? It looks something like this. The tRNA molecule has kind of a clover leaf shape. At one end of the clover leaf is kind of this exposed end where we're going to stick the amino acid. The amino acid would get stuck right there. A. And the opposite end has a region that's going to be really, really important. It's called an anticodon. Okay? So let's talk about that anticodon concept. And we've got like 3 different visions of tRNA. This is one. This is another one that's a little more accurately drawn. And this is one that's very cartoonish, with just showing the anticodon and the place where the amino acid would get stuck right there. Okay? All right. The anticodon is a sequence, and we can see it right here, of 3 nucleotides, CGG in this case. And what is true about this anticodon is it is going to be complementary to a codon on the mRNA. So if this is my mRNA strand, and these are my nucleotides, and maybe this is G, C, C, U, 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 A, G, U, something like that. Then, if I am going to have an anticodon that is going to be complementary to that, then it would have to be C, G, G. And that's what we see here. CGG is complementary to GCC. And why do we care about that? Because the tRNA and the mRNA have to have a way to communicate with each other so that the ribosome will know that, in fact, this tRNA molecule is carrying the amino acid that we really need in this location. It's carrying the right amino acid, and so that's how that communication occurs. So, if the mRNA codon is UUU, then the tRNA anticodon that would plug into that is AAA. So if a tRNA molecule comes up to the mRNA and has got GGG, then it's not a match, and it's not carrying the right amino acid, and it's not going to be allowed to bind. But if it's complementary, then it will be allowed to bond. Okay? So, Yeah, that's pretty much what we need to say. So as we translate the genetic message, then the tRNA is going to come up, and what this is showing is ribosome, large and small parts. Here's your mRNA molecule, and this is tRNA. That is the anticodon, and it's seeing if it matches. If it matches, then it will drop off its amino acid onto the ribosome, and you'll end up, in the end, after many tRNAs do this, getting a long, long chain of amino acids, and we'll make a protein. We'll show you how that happens. Now, just so you know, each tRNA has to get the correct amino acids stuck onto it, so there are enzymes that do this. So here is your tRNA molecule, here is an enzyme, and here is an amino acid that we need to build a protein, and this enzyme makes sure that the right amino acid gets stuck onto the right tRNA molecule. We have to have some ATP for that to happen, but in the end, that enzyme would make sure that this tRNA got the right amino acid. The enzymes that do this are called aminoacyl, which makes sense, amino acid, aminoacyl, tRNA synthetase. Just means we're going to synthesize or make tRNA with an amino acid stuck onto it, and that ACE tells us it's an enzyme. So there are enzymes that are in charge of that. So the whole point then of a ribosome is to make sure we are going to have a place to match up the tRNA anticodon to the mRNA codon, okay? And the ribosome, in order to do that, has 3 different positions within it. It has an A site, a P site, and an E site. A is for active. This is where a tRNA molecule would be actively matched, the codon and the anticodon. The active matching is going on here. Then we're going to have a, well, you know, I think I shouldn't say that now. We'll wait until I actually have to show you this, okay? All right, the ribosomes are made of protein, and they are made of rRNA. And it has been determined that it is in fact the rRNA that is most important in making the ribosome work. The RNA is actually acting almost like a protein in this case. It is acting like a catalyst. And I bring this up to you to remind you that ribosomes are basically one big ribozyme, RNA acting like an enzyme. Really important because RNA is, I'm emphasizing to you, is a very versatile molecule. DNA is not versatile. RNA is versatile. It can act as genetic material. It can act as enzymes. It can do a ton of stuff. All right, that's it. We will complete translation in the next podcast. All right, take care. See you next time.